Hello students, welcome to EPG Pathshala. I am Dr. Manjula Sharma from University of Delhi. Today we are going to discuss about the module Optical Microscopy, Optical Microscopy Techniques, Bright Field Optical Microscopy, Dark Field Optical Microscopy from the paper Characterization of Materials Part 1. After studying this module, you should be able to understand about the optical microscopy, in which we will study about the geometrical optics, magnification and resolving power, the depth of field, contrast. Secondly, we will study about the bright field microscopy, in which we will cover the light path, performance and advantages. Thirdly, we will study about the dark field optical microscopy, in which again we will cover the light path, its performance and advantages. First of all, let us study about the optical microscopy. Optical microscopes are also called as light microscopes and employ visible light and magnifying lenses to see tiny things. The optical microscope has been one of the most important tools in many scientific fields as well as industry. For example, biotechnology, the pharmaceutical industry, cell or microbiology, nanoscience, and microelectronics, etc. However, the resolution of optical microscope is limited by the wavelength of the light. This limitation was demonstrated by the microscopist Ernst Abbe in 1873. According to Ernst Abbe, their solution of an optical microscope is limited to roughly half the wavelength of the light, that is about 0.2 micrometers in case of visible light. It means that it is easy to distinguish whole cells as well as organelles. But things as small as a virus or single proteins are not visible through the microscope, thereby limiting the utility of this device. In this chapter, we will learn about the fundamentals of optical microscopy, including its working principle, image formation, and adjusting the microscope suitably to make use of its complete potential. Additionally, different optical imaging techniques have also been discussed. Students, now let us see the geometrical optics. A convex lens is the simplest microscope. Figure 3 shows how a convex lens produces a magnified image of an object. A light ray parallel to the optical axis of the lens passes through the focus of the lens, while a ray passing through the center of the lens does not bend at all. Figure 3 is representing the magnification of an object by a convex lens. A microscope that uses two lenses to generate the magnified image of the object is called a compound microscope. The magnified image generated by one lens is further magnified by the second lens, as shown in figure 4. Magnification of a compound microscope is the product of magnification caused by the objective and ocular, that is the eyepiece lenses. Final magnification is equal to the multiplication of the magnification of the objective and the magnification of the eyepiece. Figure 4 shows the ray optical diagram of a compound microscope. Students, now let us understand about their magnification of these microscopes. These microscopes achieve magnification by two lens system, which comprises of an ocular lens and objective. Majority of the bright field microscopes use three objective lenses which have variable magnification powers, that is 10x low power, 40x that is high power, and 100x which is almost oil immersion. The objective lens is located closer to the sample. It magnifies the sample and produces the real image. The real image is then projected upon the body tube into the oculars. The ocular again magnifies the sample by 10x and creates the final image. The total magnification achieved is the product of the magnifying power of the objective and the ocular lenses. Next is the resolving power. The magnification cannot be increased infinitely in an optical microscope because lenses are limited in their ability to distinguish two points that are very close. This property, which is called as the resolving power, is the least distance between two adjacent points in a sample which are distinguishable. Optical microscope can achieve resolutions on the order of 0.2 to 0.3 microns. Increases to a limiting value of about 0.5 microns with the oil immersion objective. This depth, which is a small fraction of the thickness of a standard biological sample, renders the optical sectioning of a sample possible. D is equal to 0.6 multiplied by lambda divided by n sine theta. 0.6 is the constant associated with the distance between two objects, which can be distinguished by the human eye as separate entities. Lambda is the wavelength of light, n is the refractive index of the medium through which the light travels, theta is equal to 1 by 2, that is the angle of the cone of light entering the objective lens. Next is the contrast. The contrast depends upon the signal to noise ratio and the physiology of vision. In this case, the signal is the image of interest and the noise is background light associated with images that are out of focus, light scattered by dirt on optical surfaces, etc. Detectable contrast depends on the ability of the eye or other measuring device to make fine distinction in intensity and color on resolvable areas of the image. Generally, the higher the noise, the greater the signal intensity needed to create a detectable image. Since cells develop little contrast in visible light, for producing detectable images, many techniques have been employed. One example of such techniques involves staining. In this regard, staining has been used for both living and dead cells. Students, now let us understand about the bright field microscopy. Bright field microscopy is the simplest illumination technique. White light is used in the bright field microscopy. In bright field microscopy, sample is illuminated from below and observed from above. The contrast of the sample in dense areas is generally caused by the attenuation of the transmitted light. It is the simplest technique due to which it is popular and widely used. In bright field microscopy, the image of the sample is the dark image on the bright background. Now, let us see the light path. The light path of this type of microscopy is very simple. No additional components are required other than the setup of the instrument. It generally consists of first, a halogen sample is used as a light source. Second, condenser lens for focusing light from source to the sample. Third, objective lens to accumulate light and also to magnify the image. Fourth, the camera to observe image of the sample. Now students, let us see about the performance. It usually has a low contrast for biological samples, but some of the biological samples absorb light to a great extent. It is useful for samples that have an intrinsic color, such as chloroplast in plant cells. It is a standard technique. Magnification is limited by resolving power. Figure 5 shows the yeast under bright field microscopy. Now, let us see the advantages and the limitations of the bright field microscopy. First, the advantages. It has simple setup and can be used for the living cells. Its limitations consist of very low contrast for most biological samples. Second, the magnification with the light microscope is around 1300x. Although higher magnifications are possible, but it is difficult to obtain the clarity of the image as the magnification is increased. Third limitation is it does not give better results for the sample which are colorless and transparent. For example, many types of mammalian cells, samples that do not have their own color can be seen without preparation. Now students, let us see the dark field microscopy. Dark field microscopy is both light and electron microscopy and uses scattered beam to obtain sample image and eliminates the unscattered beam. Consequently, field around the sample where specimen does not scatter the beam is usually dark and the image is bright. This technique is also known as dark ground microscopy. Figure 6 shows the diagram illustrating the light path through a dark field microscopy. Now, let us consider the light path. The steps, which are given below, are also described in the figure 6, where an inverted microscope is used to obtain image of the sample. First, light enters in the microscope to illuminate the specimen. Second, the patch, specifically devised disc, blocks some of the light, leaving an illuminating outer ring. Third, condenser focuses light onto the sample. Fourth, most of the light entering the sample is directly transmitted, whereas some is scattered by the specimen. Fifth, the objective lens collects the scattered light and the transmitted light misses the lens due to a direct illumination block. Sixth, therefore, image is formed by only the scattered light, whereas the transmitted light is eliminated. Now, let us consider the dark field microscopy combined with hyperspectral imaging. Dark field coupled with the hyperspectral imaging is best suited for characterizing nanomaterials. This technique has recently been used for attaching gold nanoparticles to target the CD44 plus cancer cells for the treatment of cancer. Figure 8 shows the weak beam dark field of strain around nuclear track cores. The low and high angle annular dark field imaging. In dark field technique, Images are formed with electrons diffracted into the annular aperture centered on while the scattered beam is eliminated. In a scanning transmission electron microscope for large diffracting angles, this is described as the Z contrast imaging due to the improved scattering from the atoms of high atomic numbers. Now, let us see the advantages of dark field microscopy. Dark field microscopy is a facile and efficient technique which can be employed for living biological samples, including a smear from a tissue culture, waterborne or unicellular organisms. The setup is simple, but the quality of the images of the sample is impressive. The advantages of this technique can be summarized as: first, it is an ideal technique to view unstained and transparent objects which may or not absorb light. This is because these objects have refractive indices similar to their surroundings, which makes them difficult to analyze with other techniques. Second, marine organisms like algae and plankton, diatoms, insects, fibers, hairs, yeast, and protozoa, as well as some minerals and crystals, thin polymers, and some ceramics, etc., can also be studied with this technique. Now, let us see the disadvantages of the dark field microscopy. Dark field microscopes can produce beautiful and amazing images. However, this technique also has some limitations. Major drawback of this technique is the presence of low light levels in the final image of samples. To eliminate this, illumination density must be kept very high, which, however, can damage the sample under investigation. Great care must be taken while interpreting the images produced by dark field to suppress the common dark features of bright field microscopy and vice versa. Other disadvantages include: first, dark field images are prone to degradation, distortion, and inaccuracies. Second, a specimen which is not thin enough or whose density differs across the slide may appear to have artifacts throughout the image. The preparation and quality of the slides can grossly affect the contrast and accuracy of a dark field image. Fourth, contamination is a major concern. Similarly, if oil or water is to be used on the condenser or slide, it is highly challenging to avoid air bubbles. These bubbles cause degradation, flare, and distortion of the image. Besides, they may also decrease the contrast and details of the sample. Dark field needs an intense amount of light to work. This, coupled with the fact that it relies exclusively on scattered light rays, can cause glare and distortion. So students, let us summarize what we have learned in this module. We learned about the optical microscopy, in which we covered the geometrical optics, its magnification, resolving power, the depth of field, and the contrast. Secondly, we studied about the bright field microscopy, in which we covered its light path, the performance, and the advantages. Thirdly, we studied about the dark field optical microscopy, in which again we studied about its light path, the performance, and the advantages. Thank you.